So I just wanted to say welcome and thank you very much for joining us. So this is our a joint webinar between Appleton Woods and Corning, and the topic is understanding cell culture contamination and best practices to reduce risk. Just to introduce myself, my name's Holly. And I'm the product manager for Appleton Woods, and we're I'm sure most of you know us, but just to introduce us, we're a small family-run life sciences company based in Birmingham, and we sell all sorts of life science related products, reagents, equipment, consumables, all sorts of things. I'm sure most of you are familiar with us, but another thing we like to do is to sort of get involved in the education behind some of these products to make sure that we and our customers are fully up to date on things going on in the market and best practices with things like, for example, today, cell culture contamination. So we like to it's for us as much as it is for you, because we like to learn too. And with that in mind, we've invited our close colleagues Corning along. So we've worked with Corning for many years and have a great relationship with their team, and are often out with their representatives and our representatives talking to customers, helping them, making sure that we can provide the best service and the best products from Corning for each customer. We have our colleague Raquel on the line, who is going to present the webinar today. She's the Scientific Support Manager over at Corning. So I think at that point I will hand over to her. I'm just going to stop sharing my screen, just a moment. And now should be able to hand over to Raquel.
1 second.
Oh, and I meant to say, sorry, so feel free to ask any questions. Probably the best way to do it is to pop it into the chat box, and then what we can do at the end is go through any questions that you guys have got. You can also ask them out loud if you'd prefer. So I will go ahead and hand over now. Thanks, Raquel. I'll mute myself so you don't want to hear me.
Just 1 second, everyone sees my screen?
Yep, I can see it.
Okay.
I think hopefully we're good to go. Thanks, Raquel.
Thank you. Thank you, Holly, for the introduction and for the invitation to do the webinar. My name is Raquel, as Holly says, and I'm the Scientific Support Manager for the EMEA region in Corning Life Sciences. We are based in Amsterdam, and I'm here today to have a talk about understanding cell culture contamination and the best practices to reduce the risk. During the talk, I will talk about the different agents of contamination that can occur in cell culture. The source of this contamination is also a point that I would like to spend some time on because it's very important to see where is this source coming from, and of course, in the end, measures to prevent contamination. So we will start with the agents, the different agents that can that can bring contamination to your cell culture, and that would be bacteria, yeast, fungi, endotoxin, virus, and mycoplasma. So all these 6 different agents can influence the cell culture and, of course, destroy your precious cells and experiments, and these are the things that we would like to avoid. Regarding bacteria, yeast, and fungi, there is a typo. The different sources for this kind of contamination could be lab personnel. So people, the people that are working can bring this bacteria, yeast, or fungi into the cell culture or the cell culture environment if they are not practicing the correct lab procedures. Let's say unfiltered air that may come from the laminar flow these agents can also be present in incubators if they are not properly maintained. Purified water, again, if it's not properly changed, can unfortunately can also come directly from the cell stock, and this is a problem a little bit more difficult to tackle. Also media, and of course the equipment that you use in cell culture. The detection for this type of contaminating agents would be either visual. So, for example, when the cultures appear a bit turbid, it's normal when the cultures appear with this color, because if they appear a long time, if they are in culture in a long time, the pH of the phenol red changes and it becomes orange. But the media itself is transparent. But if it's turbid, then this is an immediate sign of a possible contamination. And you can observe also under the microscope or do detection tests like microbial culture or basically culturing in an agar plate, doing a gram stain test, and I already mentioned the acidic pH and the visual. Here in this panel, you can see the cells, and also you can see a yeast growing in between the cells. So in terms of these agents, it's quite easy to spot them, especially yeast or the other types of fungi that form these very characteristic colonies radiating from the center, and it's quite easy to spot them. The defense against these agents would be, of course, if your cultures are contaminated, you have to dispose them. There is not much you can do. And as a prevention, you can use antibiotics or antifungal fungal agents. And especially on antibiotics, I will talk a bit more later on about the use of antibiotics and the concerns that also one has to have in the usage of these antibiotics, because they can also mask contamination if used for a prolonged amount of time. Another type of contamination is endotoxins, and this is a bit more complicated. Endotoxin is a lipopolysaccharide containing byproducts of gram-negative bacteria. And the common sources would be, of course, bacteria contamination, and coming from the serum that may be using in the cell culture media. There is only one method for fast detection of endotoxin, and that would be the LAL assay, which can be easily spotted here. And basically, the defense against endotoxin is aseptic techniques. That goes without saying. It's pretty much the defense against all cell culture contamination, but also sterile filtration. By sterile filtration, what we mean when we say sterile filtration is filtering a solution through a membrane that has a pore size of 0.22 or 0.2 micron. That is what is considered a sterile filtration, or of course a membrane with a lower pore size. Another one is use animal-free products, so don't use serum. This may be a possibility in some cases, but not in all cell culture. And use endotoxin specific affinity or size exclusion chromatography, and use the products from that have been through this. Another type of contamination is virus, and this is one of the most difficult to spot. Is very difficult to detect, and there is not a lot of detection methods that are practical to be used in a lab setting and in a relatively cost-efficient way. The sources is original tissues. So, for example, if you're doing cell culture that is coming from a tissue of a patient, or cells that coming from a patient, if that patient had a virus, then of course it's bringing the virus with the cells, and this is more in primary cells, or using direct tissues from a patient or an animal, whatever is the tissue coming from. If they have a virus, the virus is coming with that tissue, so it will be contaminating the cell culture. Also, the serum, if it's bought on sources that are not renowned or not going through the proper specifications before it's sold in the market, can be cross-contamination from other cultures, or from material in the laminar flow. Or it can be coming from the lab personnel. If someone comes and is working with a mild cold, nowadays, if you have a mild cold, you should stay at home and don't go to the lab. But also in the, let's call it, old days, it should also have been like that. The detection methods is basically co-cultivation, PCR, which is quite it's an easy method to do, but you, of course, you need to know which virus you are handling, because of you need to have specific primers for those. Electron microscopy is another method to detect viral detection, but is by far a very I would say, I wouldn't say tedious, but it's not so easy to do in the cell culture setting, and does not give you a very quick response. And in vivo testing. The defenses, as I mentioned before, aseptic techniques. I think this is the word that you will hear mostly throughout my talk, is aseptic techniques. Sterile filtration, even though viruses are very small particles, but it's always, if you work in sterile environment, is even the sterile filtration is always best to recommend that way. Chemical treatment, gamma irradiated serum that you can use. Use animal-free products if possible. And lastly, maintain your storage. The storage of the cells should be in the vapor phase of the liquid nitrogen. This is very important, and I will go into a bit more detail in a later phase on the talk. Of course, when you're handling when you have a virus contamination, there is also a very potential health hazard for the lab personnel that are handling. I think nowadays we are all very conscious about it. So it's always, the best is always to prevent to have a virus contamination in your cell culture, because if you don't know the source, can be a bit worrisome. And lastly, we have mycoplasm. Mycoplasm is bacteria-like microbes. They range in diameter from 0.3 to 0.8 micron, and the sources can be lab personnel, contaminated cell lines, which is one of the biggest source of the contamination of mycoplasm, media, and equipment. The detection can be done by Hooks staining, microbial culture. There is already a lot of specialized kits in the market for quick detection of mycoplasm, and of course PCR for using primers that are specific. Also, there is kits for PCR specific for mycoplasm. The defense against this contamination agent would be use, in this case, I mentioned sterile filtration, but I would like to make a small stop in parentheses here. In this case, the pore that we would highly recommend would be more 0.1 micron. And then 0.2 or 0.22. In 0.22, even though the smallest is 0.3, it depends a lot on the format of the the organism, so in terms of his morphology, and it may pass through 0.22 pores. So 0.1 would be a better option. And I also need to mention and stress that the 0.1 filtration, it does not eliminate mycoplasm, it reduces the mycoplasm, but the best way to defend from mycoplasm is not having mycoplasm. Of course, use sterile and animal-free products. In the end, lastly, storage of the cells in the vapor phase of the liquid nitrogen, and then autoclaving. Now I would like to just discuss the different sources of contamination that you encounter when you're doing cell culture, so that we are a bit more conscious about how to use them and how to prevent. The different sources of contamination for cell culture, they can come from media and serum, the usage of antibiotics, incubators, aerosols, equipment, and the liquid nitrogen that I mentioned that I will stress a bit in the end. In terms of media and serum, the problems is, so many of them can only be membrane filtration. So they can only go through sterile filtration. Why? If they have proteins in their composition, the media really cannot be autoclaved because it will destroy the proteins that are denature the proteins and other biomolecules that are in the media and that are needed for the cell culture media and the culturing of the cells. So it has to be very conscious about sterile filtration all the time. There may be some variation in nutrients, a lot variation, specifically on sera, not on media, of course. And handling of multiple cell lines and contamination of the stock. So in this situation, the best practices would be to purchase from reputable suppliers. And in the end of the talk, Holly will take over for this. So the best practices would be to buy media and sera that has from reputable suppliers, that you have a certificate of analysis, and you know exactly what it has been done and what are the tests that are done for the release of the lot, and then what the analysis and results for that specific lot. Use gamma irradiated sera. Store everything properly. So it's important to store within the correct and the recommended temperature, and also make sure that if they need to be stored away from light, that they are so. Thaw the required amount of serum. So our recommendation is always, when you receive a bottle of serum, which can be depending on the amount. This is a representation of the 500 milliliters bottle of serum FPS from Corning. The best would be if you're using every time in your culture, if you're adding 10 mLs, just create aliquots of the different serum properly labeled with a lot number, and who handled when it was thawed, so that you can just thaw once, or in this case twice. You thaw first time and then you aliquot it, store it, and then you just thaw a second time for when you are using specifically and that amount of volume. And in terms of sera, it's important that you conduct a performance testing especially if you're buying a large amount from one specific lot. Try the lot first to see that it performs as you would like your cells to perform, because as it is a biological product, there is some variability between lots. In terms of antibiotics, the problem comes from overusing the antibiotics. When you're overusing antibiotics, you mask sometimes contamination, and that leads to some relaxation on the aseptic techniques. And as you don't see the contamination, then you continue to have, you may continue to have a relaxation on these techniques, and then it will show up in the end. And the worst case scenario is that it shows up very in the end when you already have your results and you're ready to publish, and then you realize that maybe there was a contamination. Be aware that sometimes the contamination can also lead to different patterns of morphology on the cells, or different gene expression. So it is important to avoid at all costs cell culture contamination. And of course, the usage of antibiotics, like in every animal, it can lead to resistance on antibiotics. And then when you actually need to use that specific antibiotic for a specific contamination, the cells are already resistant, so you have a bigger problem in your hands. The best practices is really short-term application of antibiotics. So there is another slide coming in the end that you will see that the best is really to use antibiotics for a short amount of time, and then start reducing the amount of antibiotics for longer-term cultures. In terms of incubators, basically the incubators could be a source for fungal and chemical contamination. Why? This comes from basically a poor maintenance of the incubators. The incubators, some of them, they have, they have water reservoirs, and of course they have shelves, and this needs to be maintained and cleaned regularly. The water reservoirs can be a source for bacterial and fungal contamination, and sometimes I speak from experience. Once I entered the lab and there was literally a fungal growing on top of the water reservoir, and this, of course, fungal they expel aerosols that can go to your cultures. In terms of the shelves, that it can be unspilled non-clean media that may drop and you may not see it. So, and also the fans in the incubator can spread spores and particles. So the best practices really is to keep the maintenance of the incubator up to date, and that means just cleaning the incubator regularly, autoclaving the shelves, wash it with Lysol or 70% ethanol, and also change the water with autoclave distilled water from time to time. And of course, keeping the maintenance of the equipment itself, as in the changement of the fans or cleaning of the filters, and the same goes for laminar flows. Aerosols are also a source. So the problem is airborne particles, sneezing, coughing, dry skin, and this can lead to unsealed transportation of cells and other viruses. So the best is to use vented caps on cell culture. The vented caps, usually if you use the cell culture vessels, if you don't have vented caps, you always need to, you open in the laminar flow, you do your cell culture work, you need to close it properly, we move to the incubator, and then you need to open it again, so there is gas exchange. If you use vented caps, you avoid using, you avoid this opening and closing, or better, closing and opening again. You just close it inside the laminar flow, you transport it, and you put it inside the incubator, and there is no need to touch it again. And also the membrane in the vented caps are 0.22 hydrophobic membranes, so there is no entry of either particles or even other spills that can come from aerosol particles. And another source would be equipment. The equipment is frequently used, and it can spread contamination. And by equipment, I mean the pipettes, the pipetters that are used by different users inside the laminar flow. So the best practices is, of course, use disposables only. This goes for serological pipettes, not the equipment, of course. And the maintenance of this equipment is also important. Wipe regularly with ethanol and change the filter. The changing of the filter is very simple. All the pipetters, you can just unscrew, in this case, the orange portion, and the filter is visible. And then you just need to change it. It's also for a new filter. It's also the usage. This filter is there basically for the protection of the equipment from suctions or small drops of media or PBS that while you are using the equipment. We have all been doing cell culture, and sometimes you're there and it's a nice environment with your colleagues, and someone says something, and I've been there, and you just aspirate a bit more of your PBS, and it wets the filter. And that's okay as long as it doesn't go inside the equipment. That's why the filter is there. But as soon as the filter is wet, it's better to change it, because once it's wet, it becomes a possible source of contamination, because when you wet it, it may bring other particles that were in the liquid, and then it's dried, and then every time you put a new pipette, whatever stays there and grows there can also go into the liquid. So it's good to change it from time to time, especially if either you noticed it or you start observing that the pipetter doesn't hold the liquid anymore so well. So probably is a filter problem that is not creating is not stopping the vacuum so much. So another thing that is important to clean is the silicone gouge that is inside this portion. This is where the pipetter, the pipette, serological pipette comes in, and sometimes it may accumulate just dirt from the regular use, and it's important to clean it just with ethanol, and just when you do this procedure, just it's very simple to just do everything at once. And of course, it's important to use tips with filters for the other type of pipetters. And Corning has now released a new pipetter that can easily be autoclaved, and it doesn't have the metallic portions that the regular pipetters have that can also accumulate dirt. So it's very easy to use, and it can be autoclaved to be sterilized. And another source is the liquid nitrogen. The liquid nitrogen, the contamination usually is in the liquid phase. And this would be contamination on virus, because they can survive at liquid nitrogen, but also bacteria, mycoplasma. What happens is the cryovials, there is no cryovial in the market that is completely sealed from liquid nitrogen. It can be sealed from water, but liquid nitrogen is very difficult, even if they have the ring or so there is always, if you store it in liquid phase, there is always a portion of liquid nitrogen that can go inside the tube. And of course, if there is some contamination in the liquid phase, if it goes in, it will contaminate your cell culture or your cells, your stock of cells, sorry. So the best practices would be really to just store it in the vapor phase. This has the temperature that needs for the freezing in liquid nitrogen, and it saves you from contamination that may exist in the liquid phase, and that you don't want it to go inside your stock of cells. measures to prevent contamination. There is just one additional point that I would like to mention. This is a very representative cartoon, and that is when it comes to cell contamination. We haven't mentioned that, so because you can have stock of cells, and then with time, usually, and from experience, you have the stock of cells that came already from someone in the lab that already left the lab, and you just reuse them very often, and is very rarely that people do checks. And this is a very good representation. Everything was going along fine until they discovered that the HeLa cell line had Y chromosomes. HeLa cells come from Henrietta Lacks, and is a woman, so there is no way that she would have a Y chromosome. So it could, this can actually screw up a lot of your experiments. So a lot of the tumor cells are incorrectly labeled. There is a lot of intra-species contamination, interspecies contamination, and and cross contamination and contaminating cell lines. So this is something that you, as a lab doing cell culture, should, before starting your cell cultures, be sure that you are using the cells that you should be using. And a good way is when you purchase the cells from reputable commercial companies. But yeah. So unless you're using primary cells, it's a bit more complicated. But if you're using commercial cell lines, it's always best to start from a vial that you know what is what and what is inside. So in this, this is just a representation. You can see in lab A, this is exactly the same cell line, but in lab A, it shows the culture of these cells looks like this, and in lab B, it looks like this. And basically, in the lab A, you have not only a marker for human, but also you have some mouse cells in the culture. And in lab B, you only have human, and there is no mouse culture. So it's very important to check your cells when you are working. And one of the things that is important is doing the proper labeling of the cells. The cells have to have a unique name for the cell line. The format has to be the same, so consistent use of the same format. No synonyms. Rename every genetically engineered clones, linking it to the parental cell line. No personal identifications of the donor. So doing some authentication on your stocks from time to time is also important. This can be done by morphology or traditional methods such as karyotyping, isoenzyme analysis, or immunological methods, and other molecular methods like STRs or SNPs, and cytochrome oxidase subunit 1 barcoding. So if you do this kind of authentication, just to be sure that you are using the stocks that you should be using. And always detection, check for visible contamination, and obtain, like I mentioned, cell stocks with a certificate, such as what you can see here on the side. And in terms of management, it's also important to manage in terms of cell culture, train the personnel that are working in cell culture by not only in terms of labeling the cells that they are using or the generation of genetically modified clones, good aseptic techniques, and how to recognize easily contamination. The earlier you recognize the contamination on your cell culture, the best it is to tackle it and solve the problem as soon as possible. generate a plan of action in case of a contamination, prepare the cell banks, initiate a periodic authentication program for prevention on cell contamination and on cell culture contamination and also cell contamination. And schedule regular contamination tests, and of course schedule cleaning events for the incubators, water baths, equipment, and all the facility that uses the cell culture. So how to successfully manage cell culture contamination? It's very important to have a good aseptic technique. Like I mentioned, this is key for everything in cell culture. Strategic use of antibiotics, strategic use of the cell repository, good housekeeping by everyone, understanding the nature of the contamination, and contamination monitoring program, like I mentioned. This is an example of a cell culture log that you can have just to keep track of the cells that you have and what they should look like. And last, I just would like to mention the best practices that we have this infogram available in Corning, and that you can post it in your cell culture lab. And one of them is stick to the basics. What seems obvious to everyone may not seem obvious for a new person in the lab. So don't forget to do septic techniques, to sterile filter liquids after opening and after adding additives. So in this sense, I would like also to briefly mention that one way is also to avoid using avoid the usage that many people use the same bottle of media. So if you have one bottle of media or 3 people that are using the same bottle of media, it's important that everyone sticks to the principle that there is no serological pipettes going inside the media. So the media is poured into, for example, a 50 ml Falcon tube, and you use from that tube. So nothing goes in the media, just the media is poured inside the laminar flow in sterile conditions, and it's from that Falcon tube that you will use for your culture. Because that can be, as many users can be using the same bottle of media, it can be bringing some contamination. Another thing, sharing is caring, is it's a buzz sentence, but it's really no one likes having problems in the culture, and it's very important that if you do, especially when most of the institutes you share the cell culture room. So it's important that if someone has, this means sharing incubators as well. So if someone has a contamination, it's important to mention to the other people that use the incubator, the cell culture room, because their experiments are also in that room. So and it can be affected. So sharing is caring, and there is no one likes to have it, and no one aims to have cell culture contamination. So but it's important that if something happens, that everybody is aware and everybody can tackle and check their cultures if they are contaminated. Cover your culture. So if you need to transport the culture to minimize airborne contaminants, use cell culture labware lids, covers, or caps, vented caps, like I mentioned. Take chance out of your process. Recheck every solution sterilized in-house or stored for long periods of time. Sometimes you do a cell culture this month and you open a DMEM cell culture media, and then you're going to use it in a month's time. So check it before you use it, because you don't know how long it has been, if there was a problem with the refrigerator, if someone else used it without your knowledge. So just check the cell culture media every time you haven't used it for a very long time. Use disposable pipettes and aseptic tubing for media transfer and other liquids, or of course use an automated pipetter control that has a sterile filter to minimize the chances. But yeah. Think tidy, so wash your hands. I think we all heard a lot about washing our hands lately before putting the gloves on. Wear clean lab coats. Ideally, wear a lab coat just for cell culture that is in the cell culture room. If you're doing multiple experiments and using different facilities, it's best to have just one lab coat for cell culture. And always keep the hood clean and organized so there is no spilling, or have everything at hand's length and nothing is disturbing your movements, to reduce contaminants or to reduce dropping of media, and then you have to clean it. Simplicity is the ultimate form of sophistication. A great way is to avoid cross-contamination is keep it simple. Do not mix cell lines, bottles of media, everything inside the hood. Organize yourself and just one. If you're working on one cell line, bring everything that you need for that cell line, and then move forward with the other cell line. This will avoid cell line contamination, but also confusion. And just keeping it simple is always the best policy. And the shortcuts can become long ways ahead that you did not foresee. Again, the overusing of antibiotics is good, but it can mask some other contaminations. So just use it wisely, and always use it for initial cultures or short-term cultures, like for primary cells. But if you're going to culture for a very long time, try to reduce the amount of antibiotics over time until you have no antibiotics in the media. And then keep calm and organized. That is goes without saying, and I have been stressing that point. Test your cultures, test the in-house cell lines, and test those cell lines that have been used for years in the lab. It's never it's never bad to test them and check if they are not contaminated and if they are exactly the cells that you want to use. And be informed. Basically, information is key for everything. And with this, I finish my part. And if you have any questions, please do not hesitate to letting me know.
Thanks, Raquel. That was really good, really interesting. Yeah, so just want to hand over. Has anyone got any questions? You can either write it in the chat box, or you can stick your hand up, or you can just unmute yourself now. That's no problem. Anyone got anything they wanted to ask? Anyone? I've got one question actually that I'd thought of. One thing that we're starting to see out in the market, unfortunately, is a bit of a shortage of filter pipette tips, which obviously is going to be problematic for a lot of people's work. Do you think going forwards that with this in mind, if there is a bit of a shortage for a time, could sterile non-filter tips be just as good as long as the user is sort of using a good sterile technique?
Yes. Yes, I think so. But in that case, I would probably just clean more the pipetter itself. So we are talking about the small tips, right? The filter tip for pipetters.
Yes, of course. Sorry, yes.
So in that case, I would just clean more often the pipetter itself, maybe wipe it with ethanol, and pay more attention on while pipetting to first use the proper volumes, calibrated pipette, and just especially when it's 1 ml pipettes, when it's very fast, the suction, liquid can bounce. So it may dirty the pipetter itself. So I think in that case, there is no problem in using sterile tips that don't have a filter, but I would really pay more attention to the aseptic techniques and cleaning the pipetter more often, especially the tip where the tip goes.
Yeah, great. Thank you. That's really, really good advice. Thank you. We've got a couple of questions on the chat. Anissa, where can we access the recording of this? So what I'll do, following this webinar, I'm going to email all of you that have joined in. So what I'll do is I'll let you know then where it's going to be. We don't know exactly where it's going to be held yet, probably on our website somewhere. So I'll make sure that I'll send you the link and let you know where you can access the recording. That's a really good question.
Holly,
oh yeah.
Didn't you want to present something in the end?
Yes, I've got a slide I'll pop up at the end when we finish the questions. Thanks, Raquel. One more question is, would autoclaved pipettes need recalibration? Raquel, can you help with that one?
In principle, no. No, I think the calibration has to be done from time to time, or recalibration, or check if they need recalibration. But after autoclaving, they do not need calibration every time.
Great. Okay, thank you. Another person's access to the slides. I'll make sure that I make you aware of the recording of the webinar. I think that's all so far. Does anyone else have any more questions or comments they'd like to add? Okay. I'm just going to quickly share one more. Oh, sorry, we've got one more comment. Let me just pop that up. Okay, yeah, someone from our team has made a good point, actually, that with our own pipettes that we sell from Appleton Woods, we sometimes suggest after 10 autoclave cycles to do recalibration. I think a lot of labs probably have their own rules or guidance on this. We tend to go with after 10 autoclave cycles, just as a bit of a
a guideline. I think that's a good
brilliant. Okay, so I'm just going to quickly pop this up. So just to end on a thank you so much for your time. I know everyone's really busy at the moment, and we really appreciate you joining us. For all of those from the UK, apologies, I know there's a couple that aren't in the UK, but we will be sending out a free Corning goodie bag. So we've got Corning have very kindly sent us over some great bits like little lab timer and stationery and things like that. So what I'll be doing, as I mentioned, is I'll send out an email to everyone. In that I will let you know where the recording of the webinar is. I'll also ask you to reply with the address that you'd like your goodie bag to be sent to in the UK. Corning have also kindly given us an offer on some of their media, so on the classic media and cell culture solutions. So there's going to be an offer that you can use up until the end of the year. So what I'll do in that email is I'll also give you details of exactly which products that relates to, and a little code that you can use when you order it to make sure that when you order it through Appleton Woods, that you get the correct discount aligned. And of course, then if you've got any further questions, you can reply to me on that email as well. That's no problem. So I think that is probably everything, unless anyone has any last minute questions, or Raquel, whether you had anything else to add?
No, I just wanted to add thank you for attending and for the questions. It was very nice, and thank you, Holly, for inviting me.
Yes, you're welcome, and thank you to Raquel and to our colleagues at Corning for doing this alongside us. We really appreciate having your support with this kind of thing, getting the information from the experts. So that's all for today, guys. Thank you very much. We're going to try and put on more webinars going forward, so please keep an eye out and follow us on social media, and keep an eye on the emails, and we'll keep you updated. Great, thank you everyone.